just anchor this in terms of what we're talking about and why this matters or how this matters. So, I just opened up our favorite magazine, the New England Journal, and found this paper, which is of interest to many people around the world. How are COVID vaccines safe and or efficacious for different kinds of populations, including pregnant populations? Now, this paper was relatively small numbers of subjects, but found that COVID-19 vaccination was not associated with any of the really terrible neonatal outcomes that people might be concerned about, including stillbirth, preterm birth, spontaneous abortion, etc. So when you see this kind of report, you see this kind of original research finding, you have to confront some questions. And I like to think about these questions in the following way. Is that finding the truth? Meaning vaccination has no adverse safety effects at a population level for pregnant people? Or is it a chance finding? Is this, in other words, random error? Or could it be explained by bias? And we think about selection bias, confounding, and information bias as the major sources of bias that can contaminate our findings. Because remember, this was not a perfect randomized trial. This was observational. So here, what we're talking about today is that chance aspect or that random error. So where does that come from? That comes when we sample a population. So you'll notice that study did not take every pregnant person in the world who was vaccinated, although that is the target population here. That's the population we want to generalize our results to. But within that, we have an accessible population. Maybe it's the women who were pregnant and agreed to participate in the study. And then we have kind of a sample or a study population. Those are the people who, at the end of the day, agreed to participate and ended up in our data set. But we need to confront the fact that there are many potential samples we could have drawn. And so when we test a hypothesis, and I know you have lots of exposure to this, what we're really doing is determining the degree to which your study result can be due to chance or random error, or in other words, sampling variability. If something is statistically significant, you know it means it's less likely due to chance, although not definitively. You also know this is just not the same as something that's clinically significant. There's unfortunately or fortunately no statistic for that. That is determined based on your good clinical training, your subject matter training, to understand what kinds of differences are really clinically impactful. So let's think about sampling and the role of chance. How might this lead us to have erroneous conclusions? So imagine that we know that the truth is that maternal COVID vaccination has no impact on the baby, on neonatal, let's say, mortality risk, to be specific. Now, if we could take in our study every single pregnant person who was vaccinated and compare it to women who were not vaccinated, we would observe that truth. However, by chance alone, we could simply by random error choose a sample that would show an association between vaccination and mortality, even though we're saying that none exists in truth. And that's that kind of red dot there. So just by the nature of gathering patients, we could, by chance alone, get a cluster of patients who might have had the vaccination and also had adverse neonatal outcomes. So these are just called errors in statistical inference. We'll first, we can either get the right answer, meaning there is a true positive or a true negative, or we could get, unfortunately, the wrong answer. In other words, have a false positive where we find in our sample that the vaccine increases mortality. This is the example I just talked about, even though the unobserved truth in the population is that there is no association. Similarly, we could make a false negative error. So by chance alone, even if the truth in the population is that the vaccine increases mortality, we might fail to observe that in our sample or commit a false negative error. So beta or type 2 error is what we call the false negative error. Alpha or type 1 error is what we call the false positive error. And the degree to which we tolerate these errors in our own research are set by our own standards. So, you know that we have this sort of arbitrary threshold of alpha of 0.05. We have set that as a kind of research community